The next step in glycolysis is very much called link reaction. Or this is also known as or considered the second step in aerobic respiration. So where this reaction occurs. So definitely this is the reaction that going to takes place in mitochondrial matrix. So for the first time, the further oxidation of glucose molecule is now shifted to an organelle called mitochondria. Up to this reaction, all the conversions happened in cytoplasm. Then the further conversions of glucose is going to be occur in mitochondria. Why? Because the reactions require enzymes, and specific enzymes control a specific reaction. So basically, the enzymes required for link reaction and further aerobic respiration steps present only in mitochondrial matrix. So wherever the class of enzymes are present, there that specific reaction to be occur. So the site of second reaction or a second stage in can call the in aerobic respiration is mitochondrial matrix. And what happens during this link reaction? In this reaction, the product of glycolysis, which is called pyruvic acid, is going to be oxidized. There is a removal of hydrogens, and going to be decarboxylized, that is removal of carbon dioxide, and also it undergoes condensation with coenzyme A to form a molecule called acetyl coenzyme A. So conversion of pyruvic acid, which is a C3 compound, the key product of glycolysis, into a simple organic compound acetyl coenzyme A, which is a C2 compound, after a complex set of reactions like dehydrogenation, decarboxylation, and condensation. So the hydrogens are once again removed from the glucose residue, which is available in pyruvic acid, and further the hydrogens are taken from pyruvic acid by NAD+, which reduced into NADH+. So this is going to be called a partial oxidation and decarboxylation of pyruvic acid into acetyl coenzyme A. This is going to be catalyzed by a complex set of enzymes available only in mitochondrial matrix. So let us see this actual process of link reaction. So glycolysis, during which glucose with utilization of 2 ATP, formation of 4 ATP, and production of 2 molecules of NADH+, H+, converts into 2 molecules of pyruvic acid. So it is simply EMP pathway occur in cytosol. So as I already discussed, the further reactions of respiration shifted from cytosol to mitochondrial matrix. So mitochondria has an envelope, so let it be mitochondrial envelope, comprises to unit lipoproteinaceous membranes. From here, the pyruvic acid is going to be transported from cytosol to mitochondrial matrix. You know one more character of any lipid proteinaceous membrane, that is lipoproteinaceous membrane, which has a power of passing certain solutes on selection. So, selectively permeable, and it permits the transport of some substances through some carriers. So, here also some membrane bound carrier present in mitochondrial membrane, which is called pyruvate translocator. So, what is it? Pyruvate translocator is a membrane bound protein, serves as a carrier molecule, helps in the transport of pyruvic acid from cytosol into mitochondrial matrix. So, once the pyruvic acid enters into the mitochondrial matrix, it undergoes three important conversions, in which first it loses carbon dioxide. So, for first time in the aerobic respiration, there is a removal of carbon from glucose at link reaction. So, this is eliminated. So, this is one step, chemically decarboxylation. After that, there is a removal of a pair of hydrogen atoms from pyruvic acid, and this is called as dehydrogenation. So, this is removal of hydrogen. So, naturally dehydrogenation, otherwise what is it? Oxidation. So, second time in the event of aerobic respiration, there occurred dehydrogenation or oxidation, and hydrogens are accepted by an hydrogen acceptor called NAD plus. As a result of accepting hydrogens in terms of protons and electrons, NAD is reduced into NAD H plus H plus. So, the energetic electrons of glucose trapped once again in NAD H plus H plus. Then this pyruvic acid undergoes condensation, that is another reaction called condensation with coenzyme A. So, coenzyme A, CoA. Coenzyme A is a cofactor which condenses with pyruvic acid and finally forms a carbon compound called acetyl coenzyme A. So, a 3-carbon compound after losing carbon and undergoes oxidation and condenses with coenzyme A, converted into a C2 compound, which is acetyl coenzyme A. So, these conversions, one is what? Decarboxylation and dehydrogenation and condensations. So, a set of complex reactions occur at this stage, and all these reactions definitely biochemical reactions. So, one should expect the mediator. So, what are the mediators of biochemical reactions? None other than enzymes. So, this mega process with the 3 different reactions controlled and mediated by a complex enzyme that is called pyruvate dehydrogenase complex. So, pyruvate dehydrogenase complex is the enzyme which involves the oxidative decarboxylation and a condensation of pyruvic acid into acetyl coenzyme A. And pyruvate dehydrogenase complex is a conjugated enzyme and comprises 3 different enzymes and requires some cofactors. So, the pyruvate dehydrogenase is activated in the presence of some cofactors like thiamine pyrophosphate, TPP, and another one is called lipoic acid, and another one is magnesium, and of course coenzyme A and NAD. So, for activation of pyruvate dehydrogenase, some additional cofactors are required in the presence of NAD plus, coenzyme A, thiamine pyrophosphate, lipoic acid, magnesium. The pyruvic acid converts into acetyl coenzyme A. So, this entire process is named as the link reaction, because the product of this reaction, what is it? Acetyl coenzyme A is going to connect or link up the 2 mega events of aerobic respiration, that is glucose and Krebs cycle. So, Krebs cycle and glycolysis connected by a linking substance called acetyl coenzyme. Why? This is mainly because of a complexity of pyruvic acid structure. So, pyruvic acid cannot directly enter into Krebs cycle, but it enters into the Krebs cycle after converting into a simple form called acetyl coenzyme A. So, this is the link reaction, the second step in aerobic respiration. So, let us see the overall reaction or equation of the second step of aerobic respiration. So, what are the substrates here? The pyruvic acid. And this pyruvic acid, NAD plus, then coenzyme E. So, these are the main reactants of this reaction. Pyruvic acid, which is a C3 compound. NAD plus is a hydrogen acceptor, and a coenzyme A, another cofactor. So, this undergoes the conversion into acetyl coenzyme A, which is a C2 compound, plus CO2, CO2 is liberated, plus NAD H plus H plus. The reactants of this reaction are pyruvic acid, NAD plus, coenzyme A, whereas the products of this reaction are acetyl coenzyme A, CO2 and NADH plus H plus. Basically, after glycolysis, the participation of 1 molecule of glucose, we will get 2 molecules of pyruvic acid. So, for conversion of 2 molecule of pyruvic acid, they requires 2 molecules of NAD plus and 2 molecules of coenzyme A. As a result, we will get 2 molecules of acetyl coenzyme A. So, there is CO2, 2 molecules of CO2 liberation and 2 molecules of reduced coenzyme. You can call this as a reduced coenzyme. Two molecules of NAD, H plus, H plus. With the mediation of an enzyme, pyruvate dehydrogenase complex. So, pyruvate dehydrogenase complex. So, this is the reaction or the summary of second step of aerobic respiration. The next event of aerobic respiration. So, that is also going to be occur in mitochondrial matrix. So, after the EMP pathway in cytosol, we just witnessed a simple equation or process in mitochondrial matrix involving conversion of pyruvic acid into acetyl coenzyme A. And this acetyl coenzyme A, which is a 2-carbon compound, a residue of glucose, is going to be participate the next event, which is called Tca cycle, otherwise a Krebs cycle. So, the so-called end product of link reaction, that is called acetyl coenzyme A, going to be participate in a cyclic pathway called Tca cycle. In a Tca cycle, the acetyl coenzyme A going to undergo complete oxidation and decarboxylation. So, that cyclic pathway of complete breakdown of acetyl coenzyme A to release CO2 and water is called tricarboxylic acid cycle, otherwise called Krebs cycle. So, during which there are number of compounds are formed, say X, or rather K, L, M, N, O, P, like that. So, number of intermediate compounds are formed during the complete oxidation where hydrogens are removed, and a complete decarboxylations where CO2 are eliminated, and formation of water. So, complete oxidation, decarboxylation of acetyl coenzyme A in a cyclic manner is called Tca cycle or Krebs cycle. It is also going to be occur in mitochondrial matrix. So, the link reaction and Krebs cycle both going to be occur in mitochondrial matrix. So, what are the compounds that are formed? To be brief, the acetyl coenzyme A conversion starts with the participation of a compound called oxaloacetic acid, and which combines with acetyl coenzyme A forms a citric acid, later cisaconitic acid, further isocitric acid, then oxalosuccinic acid, then alpha-ketoglutaric acid, then succinyl coenzyme A, followed by succinic acid and a formation of fumaric acid, malic acid, and then oxaloacetic acid. So, this is a metabolic pathway which occurs in a cyclic manner and conditioned by a set of reactions. So, it is called Krebs cycle or Tca cycle. So, we will discuss this entire pathway in detail. So, let us start the details of Tca cycle. So, Tca cycle is otherwise called tricarboxylic acid cycle. Tricarboxylic acid cycle. Why it is called tricarboxylic acid cycle? Because during this metabolic pathway, there form number of intermediate acids, of which the first 4 acids have 3 carboxylic groups. So that means 3 COOH groups. So those carbon compounds with 3 carboxylic groups, one is called citric acid and another one is called cis aconitic acid and another one is called isocitric acid and another one is called oxalosuccinic acid. So the first 4 acids that are formed during this cyclic pathway of complete oxidation and decarboxylation coenzyme called tricarboxylic acids. So citric acid, cis-aconitic acid, isocitric acid and oxalosuccinic acid. Hence, this pathway named as a tricarboxylic acid cycle, and it is also called citric acid cycle. Citric acid cycle. So why this is called citric acid cycle? Again, during this complex metabolic pathway, there are a number of organic acids are formed as intermediates, of which the first formed acid is a citric acid. So the first formed stable compound during this pathway is a citric acid. Hence, this pathway named as citric acid cycle. And this is the pathway which is also called as organic acid cycle. Why? Because again, during this conversion of acetyl coenzyme into CO2 and H2O, they formed a good number of organic acids. Hence, it is called organic acid cycle. But very popularly, Tca cycle is called Krebs cycle. So Krebs cycle is a very common popular name to the Tca cycle. So again, why? Because all the reactions that involve the complete oxidation and decarboxylation of acetyl coenzyme into water and carbon dioxide were discovered and explained and presented by a great scientist called Sir Hans Krebs. Sir Hans Krebs. So that's why, to honor the discoverer of this pathway, discoverer of this metabolic event, this process very much named as Krebs cycle, just like the glycolysis. And glycolysis process was discovered by 3 scientists. Who are they? Emden, Meyerhoff, Furness. Is it right? So to honor the discoverers, the glycolysis named as EMP pathway. Similarly, to honor the discoverer of Tca cycle in living cells during aerobic respiration, named as Krebs cycle. And the location or the site of Krebs cycle is mitochondrial matrix. And in Krebs cycle, what happened? There is a cyclic series of reactions conditioned by a set of enzymes. So it is a cyclic pathway. But the so-called EMP pathway is a linear pathway. That is not a cyclic pathway. You know that metabolic pathways in a living cell either linear pathways or cyclic pathways. There are a good number of examples to support these 2 statements. One example is a linear pathway, our EMP pathway, and a cyclic pathway, is the so-called Krebs cycle. So what happens here? Chemically, this Krebs cycle involves what? Complete oxidation and decarboxylation of acetyl coenzyme A into water, carbon dioxide. So this is the pathway. Once again, oxygen is not directly involved. So no direct utilization of oxygen during Krebs cycle, but the glucose energy is very much liberated during Krebs cycle, so that is stored in NADH plus H pluses and also FADH2s. So at the end of Krebs cycle, you will notice liberation of CO2, formation of NADH plus H plus and FADH2 and water. So this is the introduction part of Tca cycle. So we will discuss the equations, the details of Krebs cycle in equations. So during the Krebs cycle, the acetyl coenzyme going to be condensed with oxaloacetic acid. So actually this is a process by name condensation, during which the acetyl group in acetyl coenzyme, actually it is acetyl group is a residue of glucose. It is now supposed to be undergo complete oxidation and decarbonation, but it itself cannot undergo oxidation and decarboxylation. It requires the support of another compound that happens to be oxaloacetic acid. So during this first reaction of Krebs cycle, there is a condensation of acetyl group of acetyl coenzyme with oxaloacetic acid. Oxaloacetic acid is a C4 compound which is present naturally in mitochondrial matrix, which is reacts with the acetyl coenzyme A and converts into the compound called citric acid. So the first step in aerobic Krebs cycle is a condensation of acetyl coenzyme A with oxaloacetic acid to form citric acid. During this process, the coenzyme A is liberated, of course, which is available for further next conversion of one more pyruvic acid, and during which one molecule of water also utilized. So OAA is a prepared substance in mitochondrial matrix taking part in the oxidation and decarboxylation of the acetyl coenzyme. So on condensation, acetyl coenzyme forms citric acid. So basically the two reactants of this reaction, one is a C4 compound, another is a C2 compound. So one would expect a compound having 6 carbons. So C6 carbon compound called citric acid. So as already discussed, the first formed compound among the different intermediates of Tca cycle is citric acid, hence it is aptly named as citric acid cycle. And the enzyme for this reaction is citric synthetase. So citric synthase is the enzyme which catalyzes the first reaction. So citric acid now undergoes some characteristic changes which consider to be isomerization in a couple of reactions. That means with the two simultaneous reaction, citric acid first converted into cis aconitic acid by losing water molecule, and immediately it converts into isocitric acid with the addition of water. So, chemically that citric acid into isocitric acid conversion we consider as isomerization, where there is a removal of water and immediately addition of water. So, citric acid, the product of first reaction of Tca cycle, is now you can straight away call isomerized into isocitric acid. Both these reactions occurs simultaneously in the presence of same enzyme, which is called aconitase. So, aconitase is the enzyme which involves the conversion of citric acid into isocitric acid. So, two reactions finally convert citric acid, a C6 compound, into isocitric acid. So, take the carbon count. A C6 compound just loses water and again gains water. So, there is no removal of any carbon, there is no addition of any carbon atom. So, the number of carbon atoms going to be same in cis aconitic acid. So, it is a C6 compound, and isocitric acid is also a C6 compound. So, citric acid, cis aconitic acid and isocitric acids. Now, the isocitric acid is converted into another compound called oxalosuccinic acid. So, oxalosuccinic acid, I will write the total name, oxalosuccinic acid, and this conversion of isocitric acid into oxalosuccinic acid occurs after the removal of hydrogens. So, first time in Krebs cycle during aerobic respiration, there is a liberation of hydrogens from isocitric acid in order to convert into oxalosuccinic acid. So, this is a dehydrogenation reaction where the hydrogens, which are hydrogen atoms, contains 2 protons and 2 electrons. So, the redox equivalents in the form of hydrogen atoms removed during the conversion of isocitric acid into oxalosuccinic acid, and these hydrogens actually belongs to glucose accepted by NAD plus, because NAD plus is a good hydrogen acceptor and it converts into NAD H plus H plus. So, what happened here? Isocitric acid is oxidized and NAD plus is reduced. Isocitric acid is oxidized into oxalosuccinic acid and NAD is reduced into NADH plus H plus. So, this is the process catalyzed by an enzyme called isocitric dehydrogenase. So, isocitric dehydrogenase is the enzyme which catalyzes conversion of isocitric acid into oxalosuccinic acid. So, here all hydrogens are removed, but not carbons, hence oxalosuccinic acid is one more C6 compound. And this oxalosuccinic acid converts into alpha ketoglutaric acid, alpha ketoglutaric acid after removal of CO2. So, in Krebs cycle, of course, so in mitochondrial matrix, this is the second time the carbon dioxide is liberated. First time during the conversion of pyruvic acid into acetyl coenzyme in link reaction, and the second time during the conversion of oxalosuccinic acid into alpha ketoglutarate acid. So, what should be it is chemically? Decarboxylation. So, dehydrogenation followed by decarboxylation, and this is catalyzed by oxalosuccinic decarboxylase. Oxalosuccinic decarboxylase is the enzyme that catalyzes this reaction. So, oxalosuccinic decarboxylase. So, as a result of carbon removal, the next formed compound going to maintain only 5 carbons. So, alpha ketoglutarate acid is one of the respiratory intermediate formed during Tca cycle having 5 carbon atoms. So, this is somewhat an important compound going to be asked frequently, that is the number of carbon atoms present in alpha ketoglutarate acid. 5. The only 5 carbon compound to be formed in Krebs cycle is alpha ketoglutarate acid. Next, the alpha ketoglutarate acid undergoes a complex set of reactions almost similar to the link reaction. So, already you can see in the link reaction in characters, there is a decarboxylation, there is a dehydrogenation, there is a condensation. Similarly, here also alpha ketoglutarate acid undergoes decarboxylation and it undergoes dehydrogenation. So, once again a pair of hydrogen atoms are being removed from alpha ketoglutarate acid and condensation with coenzyme A. So, decarboxylation, dehydrogenation and condensations occur during the conversion of alpha ketoglutarate acid, and that product is succinyl coenzyme A. Succinyl coenzyme A is formed from alpha ketoglutarate acid after what? Decarboxylation only, condensation only, dehydrogenation only, all the above. So, definitely all the above. There is a complex conversions occur between alpha ketoglutarate acid into succinyl coenzyme. So, these hydrogen atoms are taken by again NAD pluses and which are reduced into NAD H plus H plus. So, once again during the aerobic respiration there is an hydrogen liberation accepted by an hydrogen acceptor NAD plus which forms NAD H plus H plus. So, notice one thing, we are discussing about a reaction or a process which is mainly meant for production of ATP. So far we have finished more than 15 reactions, but only in the initial steps, that is in glycolysis, we witnessed generation of ATP directly, but after that neither in link reaction nor in the off part of Krebs cycle no ATP generation, but the energy of glucose is removed in the form of electrons at link reaction once, and during conversion of isocitric acid into oxaloacetic acid, and during conversion of alpha-ketoglutaric acid to succinyl coenzyme A. So, those high energetic electrons of glucose are trapped and stored in NADH plus H pluses. So, still the energy of glucose is stored in different molecules like NADHs, where they are further undergo some reactions. That electron energy is utilized for ATP synthesis. We will discuss later. Then coming to this enzyme, that is the enzyme that control this reaction is alpha-ketoglutaric dehydrogenase, alpha-ketoglutaric dehydrogenase complex, since a multiple set of reactions occurred here. So, alpha-ketoglutaric dehydrogenase complex is the enzyme. Now, coming to the succinyl coenzyme, because of the loss of carbon from alpha-ketoglutaric acid, which is a C5, then there forms a C4 compound, succinyl coenzyme A. So, starting with a C6 compound and coming to a C5 compound after little conversion, again a C4 compound. In fact, the number of carbon atoms in all these intermediates going to be an important item as far as MCQs are concerned. So, the first and last C5 compound in Krebs cycle is definitely alpha-ketoglutaric acid. Then succinyl coenzyme A converts into a succinic acid. During this process, the succinyl coenzyme undergoes cleavage, that means breaking, and during that bond breaking, some energy is liberated and a coenzyme is released, so which is made available for next reaction. And the energy liberated during this conversion involves in the formation of a bond between ADP and IP to form the energy currency. So, for the first time in Krebs cycle, we are seeing the formation of cell currency, that is ATP. So, in plant cells, during the conversion of succinyl coenzyme A into succinic acid, there is a direct production of ATP, which is called SLP. Already you have seen that SLP steps in glycolysis. So, this is SLP step in Krebs cycle. In plants, at this place, ATP is synthesized. If it is an animal, in animal cells during Krebs cycle, GTP is first formed, which is a form of energy currency, but not a common usage. GTP, guanosine triphosphate. So, in animal cells, during the cleavage reaction of Krebs cycle, GTP is formed, which later converts into GDP and involves in ATP synthesis. So, GDP or GTP is formed during Krebs cycle of the animal cells. Later it converts into ATP, but if it is a plant cell, straight away ATP is formed. This reaction is catalyzed by an enzyme called succinyl thiokinase. Succinyl thiokinase is the enzyme which involves the SLP step in Krebs cycle. Then succinic acid is a C4 acid which converts into fumaric acid. So, fumaric acid is formed from succinic acid, during which hydrogens are removed. So, we can say that succinic acid is now oxidized into fumaric acid after losing a pair of hydrogen atoms, which are taken by another coenzyme, that is FAD. So far, in these reactions of hydrogen dehydrogenase reactions, NAD acting as hydrogen acceptor, for the first time flavin adenine dinucleotide acts as hydrogen acceptor and reduced into FADH2. So, this is one more type of reduced coenzyme holding the energetic electrons and protons of glucose for a while. So, FADH2 is formed during the conversion of dash into dash, that is a 4-carbon compound succinic acid into fumaric acid in the presence of an enzyme succinic dehydrogenase. Succinic dehydrogenase enzyme. Succinic dehydrogenase enzyme. So, fumaric acid is a C4 acid, and this is a special enzyme because of its location. In general, all the enzymes required for Tca cycle to be present in mitochondrial matrix, but the one enzyme of Tca cycle differ from others in its location, that is succinic dehydrogenase. Instead of presenting in matrix, it located in the inner membrane of mitochondria. Inner membrane of mitochondria. So, it is straight away you can say membrane bound enzyme, succinic dehydrogenase, which catalyzes the oxidation step. Number in Krebs cycle is the third. Third oxidation step in Krebs cycle is catalyzed by succinic dehydrogenase, and fumaric acid converts into malic acid, which is also C4 compound, where water is added. So, there is an addition of water, another molecule, to fumaric acid to convert into malic acid. So, it is chemically what? Dehydrogenation, hydrogenation, hydration or dehydration. So, simply hydration, and enzyme is fumarase. So, fumarase is the enzyme which converts a fumaric acid into malic acid. So, finally, the C4 compound malic acid is regenerated into OAA or converted into OAA after losing hydrogens. So, in the last step, malic acid undergoes oxidation into oxaloacetic acid, and these hydrogens in this aspect taken by NAD pluses converts into NAD H plus H plus. So, this is once again oxidation step, dehydrogenation, and a C4 malic acid forms into oxaloacetic acid. So, oxaloacetic acid is regenerated and is ready for conversion of one more acetyl coenzyme A in a cyclic manner. So, in this case, the acetyl group of acetyl coenzyme is totally decarboxylated at two places. This is one CO2 and another one. So, the two carbons in acetyl coenzyme are just liberated, breaking of carbon-carbon bond, and hydrogens of acetyl coenzyme also released. So, total oxidation of acetyl coenzyme A in a cyclic manner is completed. For continuous occurrence of this pathway, there is continuous supply of OAA is mandatory. If OAA is not available, then there is no further conversion of or oxidation and decarboxylation of acetyl coenzyme. So, it is a mandatory one. That means OAA regeneration is as important as the aerobic cyclic pathway in mitochondria. So, OA must be regenerated. So, this is called citric acid cycle. Coming to the overall reaction of Tca cycle. So, what are the reactants and what are the products? So, this equation says that the acetyl coenzyme A plus another reactants NAD pluses and FAD plus and water converts into CO2 plus water plus NADH plus H plus FADH2. FADH2, and in this case there is an ADP also, ADP plus IP. So, ATP is formed. So, one molecule of acetyl coenzyme participation in Tca cycle, they require 3 NAD plus and 1 FAD plus, and they consume 3 water molecules, and 1 ADP forms 2 CO2 and 1 H2O and 3 NADH pluses, 1 FADH2 and 1 ATP. So, in one turn of Tca cycle, one molecule of acetyl coenzyme, but actually there are 2 acetyl coenzymes. So, those 2 acetyl coenzymes involves in Tca cycle, then 2 acetyl coenzymes, 6 NAD pluses, 2 FADH2, 6 water molecules, 2 ADP and inorganic phosphates, 4 CO2, 2 H2Os and 6 molecules of NADH and 2 molecules of FADH2 and 2. So, overall reaction of Krebs cycle is like this. And in Krebs cycle, CO2 molecules are, in Krebs cycle oxygen is, in Krebs cycle ATP production or ATP molecules are, and in Krebs cycle NAD pluses, FAD pluses are going to be, what happens? So, the CO2 molecules are liberated, already decarboxylation reactions, and oxygen molecules not utilized. So, still there is no participation of the main substance for aerobic respiration, oxygen. ATP are produced through SLP. NAD pluses are reduced into NADH plus, H pluses, and FAD pluses are also reduced into FAD H2s. So, this is a little summary of Tca cycle. So, final, the total list of reactions that occur in this Krebs cycle. There are 10 reactions, 10 different enzymes and products, chemical nature of all those reactions. So, first one is a condensation reaction catalyzed by citrate synthetase. Second one is dehydration reaction catalyzed by aconitase. Third reaction is chemically hydration catalyzed by aconitase, and this one oxidation. The oxidation is here first in Tca cycle, second in mitochondrial matrix, third in aerobic respiration, where enzyme is isocitric acid dehydrogenase. After that, there is a decarboxylation, then again oxidative decarboxylation. So, you can call this oxidation step, that is sixth reaction in Krebs cycle. Chemical is oxidative decarboxylation. In sequence is the second oxidation in Tca cycle. See, this is the first one, the second. So, that means third oxidation in mitochondrial matrix, that considering link reaction, and altogether fourth oxidation step in aerobic respiration, followed by cleavage, otherwise called substrate level phosphorylation. So, the so called seventh step in Tca cycle is SLP, enzyme is succinyl thiokinase, and the next one is oxidation, and this eighth step is an oxidation. Actually, this is third oxidation in Tca, fourth oxidation in mitochondria, and fifth oxidation step in aerobic respiration catalyzed by succinic dehydrogenase. Next reaction is hydration, where water is utilized, and last and final reaction in Krebs cycle is once again in oxidation. So, it is the last oxidation in aerobic respiration, otherwise sixth oxidation step in aerobic respiration. Already we noticed and discussed the number of oxidation steps in aerobic respiration, sixth. So, last oxidation step in aerobic respiration catalyzed by an enzyme malic dehydrogenase. So, correct sequence of enzymes and a correct sequence of chemical reactions that occur during the Krebs cycle. Okay, to conclude this Krebs cycle, let us see some questions on Krebs cycle. So, in terms of the NEET, so first question, read it. Pick out the incorrect statement with reference to aerobic respiration. I have given 4 statements. Identify the incorrect, that means wrong. So, last reaction of Tca cycle is an oxidation reaction. Certainly, yes. First reaction of citric acid cycle is a condensation. This is also true. And third reaction, penultimate. Penultimate means last but one. The last but one reaction of Krebs cycle is a dehydration. So, remember what is the last but one reaction? Conversion of fumaric acid into malic acid, where water is added but not removed. So, that is a hydration but not dehydration. So, this must be the answer. Let us see the fourth one. Second and third reactions of Tca cycle are catalyzed by aconitase. This is also true. So, after reading all these 4, the correct answer, that is incorrect statement regarding aerobic respiration is the third one. It is not dehydration but hydration. Okay. See the second one, number of NADH plus H pluses and ATP and FADH2 molecule that are formed during one turn of Krebs cycle respectively. So, here you must concentrate on turn, number of turns of Krebs cycle. If it is given one turn, that means one molecule of acetyl coenzyme is participated. So, recall the cycle. During one turn of Krebs cycle, how many NADHs, how many FADH2s and how many ATPs are formed. So, given 6 NADH2s, 2 ATPs and 2 FADHs. I think it is not correct. 6 NADHs, 1 ATP and 2 FAD. This is also wrong. 3 NADHs, 2 FADH2s or 2 ATP, also wrong. But there are 3 NADHs 1 ATP and 1 FADH2. So, answer is 4. If it would be 2 turns of Krebs cycle, then answer going to be the so called 6, 2 and 2. So, there is a trap. And last one, the first and last enzymes of Krebs cycle respectively are. So, what is given? The first reaction enzyme, the last reaction enzyme. Already we have studied the tabular form. So, by taking that assistance of the tabular form, we can answer. Citrate synthase and fumarase. This is wrong. Aconitase and malic dehydrogenase is a partly correct, but not totally correct. Citrate synthetase, of course, first enzyme. Malic dehydrogenase, definitely the last enzyme. So, this must be the answer. For confirmation, you are supposed to read all 4 options at any cost. So, read the 4th one, even if you identify the 3rd one is right. Who knows, maybe a trap. So, 4th one is given as a malic dehydrogenase and a citrate synthase. Both are similar, but what is here respectively? That means the first one member is concerned with the first enzyme, last member of the pair concerned to be the last reaction, but what is given here? Reverse. So, finally, the 3rd answer is the correct one.